Hello and welcome to the new lecture series. I will be starting with the metabolism of carbohydrates, and we will be seeing various major and minor metabolic pathways. But before starting with the major and minor metabolic pathways, first let's see how it is digested and how the carbohydrates are absorbed. So we will start with the digestion and absorption of carbohydrates in this lecture. So carbohydrates which you take in your diet, they are mostly in the form of polysaccharides. So in digestion, what the body does or what the digestion does is it breaks the polysaccharides into monosaccharides into simpler forms so that those monosaccharides can be absorbed properly. So if I start with the digestion process, you take food mainly in the form of polysaccharides. The main polysaccharide which you take in your diet, as you know very well, is starch, in the form of starch. And if you see the structure of starch, it is present in two forms. It is present as amylose and it is present as amylopectin. And you know very well the digestion process while eating, it starts in mouth itself. So digestion is starting in your mouth itself. In your mouth, when you chew the food, the salivary glands secrete salivary alpha amylase. So there is an enzyme which is called as salivary alpha amylase, which initiates the digestion process of polysaccharides. And if you see the structure of amylose and amylopectin, this amylose is a linear structure, whereas this amylopectin is a branch structure. It has got branches. This is amylopectin and this is your amylose. So what this salivary alpha amylase will do, this salivary alpha amylase, it has got few functions, like it will break the alpha 1,4 glycosidic linkages. So while the food is being chewed in your mouth, the salivary alpha amylase will break the alpha 1,4 glycosidic linkages which is holding the glucose monomers, and it will break it into, like suppose the salivary alpha amylase, suppose it is working on this glycosidic linkages, or it might work on this glycosidic linkages, so it might produce maltose, it can produce maltotriose, it can produce isomaltose which has got alpha 1,6 linkage, or it can produce various oligosaccharides which are also known as dextrins or limit dextrins. So this is maltose, this is isomaltose, this is maltotriose, this is maltotriose and this is isomaltose. This isomaltose has got alpha 1,6 linkages, whereas the maltose and maltotriose has got the alpha 1,4 linkages. So these constituents can be produced in your mouth during the digestion process. The digestion itself is starting in the mouth. But due to very short stay of food in your mouth and due to the very low activity of salivary alpha amylase, around 20 to 30% of digestion only occurs in the mouth. Only around 20 to 30% of digestion, or around 20 to 30% of polysaccharides are broken down in your mouth itself. Okay, rest digestion is not occurring. So after mouth, your food reaches stomach. Okay, but as the food reaches stomach, due to the low pH of the stomach, there is no digestion of carbohydrates. No digestion of carbohydrate is occurring in the stomach due to low pH, because at that pH of around 2 to 3, the salivary alpha amylase, which is wrapped around the food bolus or food particle, it stops its action due to the acidic nature of the stomach. So there is no digestion in the stomach. After stomach, the food reaches the small intestine. Or you can say food reaches the duodenum. As the food reaches the duodenum, the food bolus or the food particle is surrounded by acidic environment. So the food bolus is acidic. This acidic nature of the food bolus, first it has to be neutralized. It has to be neutralized. So for neutralization, your pancreas secretes some digestive juices, like it secretes bicarbonate, and your liver secretes bile. So due to presence of secretion of bile and bicarbonates, this acidic bolus becomes neutral. Okay, so this neutralization of this acidic chyme, now this neutralized bolus will undergo further digestion in the presence of another enzyme, which is pancreatic alpha amylase. This pancreatic alpha amylase has got the same action as the salivary alpha amylase, means it will break the alpha 1,4 glycosidic linkages. It will also break alpha 1,4 glycosidic linkages, and it is more powerful. It is more powerful and in action as compared to salivary alpha amylase. More powerful means it will break all the alpha 1,4 linkages remaining. Remaining alpha 1,4 linkages will be properly broken by the pancreatic alpha amylase. So similarly, you will get maltose, you will get maltotriose, you will get isomaltose, and some oligosaccharides also. This is maltose, maltotriose, and isomaltose. So as you can see, these products of digestion are right now in the duodenum of the small intestine. Now this broken down products will, from duodenum, it will go to jejunum. In jejunum, it will reach to proximal part of jejunum, where certain special enzymes are there which are known as disaccharidases. Certain special enzymes are there in the proximal jejunum which are known as disaccharidases, and they have a very specific location of these special disaccharidases. Suppose this is a structure of a lumen of small intestine. This is your jejunum area, and these are, suppose, is the intestinal cell. These are various intestinal cells in the proximal jejunum. These red structures are the intestinal cells. Now in these intestinal cells which are located in the jejunum, there are special disaccharidases which are located in the apical part of this jejunum. These green structures, as you can see, these are the special disaccharidases. Now these special disaccharidases, it can be maltase, it can be lactase, it can be sucrase, it can be isomaltase, or it can be certain oligosaccharidases. Okay, these are certain enzymes, special disaccharidases, which are located in the apical part of the small intestinal cell. Now, as you can see, the products of digestion were maltose, maltotriose, isomaltose, and certain oligosaccharides. So all these will be broken down into constituent monosaccharides in the presence of these disaccharidases. Like maltase enzyme will break down maltose into two glucose molecules. Sucrase enzyme will break down sucrose. Sucrase enzyme will break down sucrose into glucose and fructose. Lactase enzyme will break down lactose into glucose and galactose. Oligosaccharides will be broken down into n number of glucose units. Okay. Similarly, all the products of digestion will be converted into simpler monosaccharides. Those simpler monosaccharides, it could be glucose, it could be fructose, or it could be galactose. So the products of digestion, you will see they are all monosaccharides: glucose, fructose, and galactose. These are the major products of digestion. And if you see the final products of digestion, more than 80% product would be glucose, and less than 10% will constitute galactose and fructose. Okay. So glucose, galactose, and fructose would be the end product of digestion. Now after digestion, after complete digestion, they need to be absorbed. They need to be absorbed. And if you see the rate of absorption, will be always higher for hexose sugar when compared to pentose sugar. The rate of absorption would be higher for hexose sugar when compared to pentose sugar. And in pentose sugar also, and in hexose sugar also, you will see the rate of absorption would be higher for galactose. Galactose, which is a hexose sugar, it has a higher rate of absorption as compared to glucose and fructose. So galactose will have the highest rate of absorption, followed by glucose and fructose. After fructose, it would be pentose sugar. So the rate of absorption you see is highest for galactose, followed by glucose, fructose, and pentose sugar. Now the main thing is how these galactose, glucose, and fructose, all these are absorbed from the intestine. This we will see in the next lecture. Thank you.